Thanks very much, Mihai. And yeah, it's a shame I can't be there in person. It looks like a great session, but yeah, I look forward to seeing the rest of it online. Okay, so firstly, what do we mean by genome-resolved metagenomics? Well, essentially, it's just trying to obtain genomes directly out of microbial communities from mixed DNA samples. And early on in DNA sequencing, this was considered a very challenging thing to do, almost impossible. But through some of the statistical methods that I'll talk about in a bit, yeah, we've made a lot of progress, and it's transformed our understanding of microbial diversity. So this is nicely illustrated by this review paper in 2016. So this is basically the tree of life. So eukaryotes is this little bit over here, that includes us. And the key thing is every red dot corresponds to a phyla for which at that point there was no cultured representatives. So these are organisms that we know about through genome-resolved metagenomics, but that we can't actually grow in the lab. So you can see you've got this vast diversity of, say, the candidate phyla that at that point were completely, unculturable. So it really has transformed our understanding of microbial diversity, but this was all based on short-read metagenomics, and that has limitations that I'll give a brief introduction to. What I'll talk about in the main part of my presentation is what we can do in terms of long, accurate reads, and in particular, I'll present an assembler that we've developed for hi-fi reads that enables you to get the majority of the community as high-quality genomes, as I'll explain. So we published that earlier in the year, and then I'll conclude with a little bit of a sort of future outlook in terms of what we're likely to be able to do with Oxford Nanopore reads. Okay, so just a very, very brief introduction to the microbiome and metagenomics, which I think will be useful for the rest of the session. But the key point is, in the analysis of the microbiome, we have a whole community of different species present. So we take a sample and we extract DNA. And so if we're doing shotgun sequencing, it means that all those different bits of DNA can come from a different species, and we don't know a priori which species a piece of DNA comes from. So in terms of sequencing data, what is available today is we have short, low error reads, and that is the vast majority of data sets, in fact. For the last few years, we've had long, so over 10 kilobase pair, but highly erroneous reads, somewhere up to about 5% error rate. The latest Oxford Nanopore sequence data is a lot better than that. It's about a 0.5% error rate. But what I'm going to focus on today is long, accurate reads. So this is represented by Hi-Fi PacBio, and there you've got long reads and very low error rates, somewhere like 0.01 or 0.1%. Another form of information we may have is multiple data points from the same samples, so longitudinal time series. And I'll talk very briefly about something called Hi-seq. So this is it's not a different sequencing technology, it's a different library prep, where you basically cross-link physically co-located DNA and sequence across the cross-links. And what that gives you is it's the only way to do in situ metagenomics, which potentially links, say, extrachromosomal elements like plasmids and phages into genomes. And this is actually all nicely reviewed by a recent review in Nature Genetics earlier this year by Army Bart and co-authors. Okay, so just briefly to give you an overview of short-read metagenomics assembly, and I apologize, this is very simplistic. It represents my understanding of it. But essentially, assembly is just about finding overlaps. We've got two reads, the overlap here, and we assemble them into a longer sequence. And there are two paradigms for assembly: overlap layout consensus, which is also effectively the same or similar to string graph assembly, and de Bruijn graph assemblers. Now, in OLC, this is sort of how a human might intuitively assemble reads. You compare each read to every other one, and you look for overlaps. And then you form a graph where each node of the graph is a read, and the edges are overlaps to the other reads. And then we're looking for high confidence paths through that graph. When we've found one, we may collapse the reads onto that path to form a consensus sequence. The problem with this is it scales very badly with read number because you have to do that all versus all comparison. You can use minimizers, which is something I'll talk a little bit about in a minute to speed up that comparison. But essentially, it's computationally intensive when you have large read numbers. So a big breakthrough for short read assembly was de Bruijn graphs. So in a de Bruijn graph, your first step is to decompose reads into k-mers, so short nucleotide words, and then you build a graph out of those k-mers, and you join. So here's an example. So we've got a read of 6 nucleotides and another read there. We form our graph. In this case, we're using trimers with each of those overlapping k-mers, and we put an edge in if the k-mers are present in the same read, and then we repeat that for the second read. And the sort of magical thing is you find the overlap between those reads. So in this way, you can find overlaps without actually having to do any alignment, explicit comparison. Now, the overlap length is defined by that k-mer length, which is fixed, which is one of the drawbacks of de Bruijn graphs. But the big advantage is the overall complexity scales with the complexity of the genome, not the read number. Now, the optimal overlap length will be a function of coverage depth. If you have high coverage genomes, you want to use k-mers that approach the length of your reads, because if you think about it, you've got a good chance of observing a long overlap. And the longer the k-mer you use, the better you are at resolving repeats. But for low coverage genomes, you want to use a small k-mer length because you'll only see small overlaps in that case. So another big breakthrough in short read assembly was iterative k-assemblers, things like IDBA, UD, Spades, and the one we typically use now, MegaHit. These have the advantages of the best of all worlds in terms of those k-mer lengths. And then assembly just consists of processing those graphs. For instance, we might remove tips, or which is shown in red here, or bubbles, which will correspond to variants, and then we output the long contiguous regions, stretches of nodes that have one input and one output as our contigs. So that's a very basic introduction to de Bruijn graph assembly. Now, when we apply it to metagenomics, we have additional challenges. So just as with genomes, we'll have repeats within genomes, but in metagenomics, because of strain variation, we can have very long repeats between multiple strains of the same species. And on top of that, those species will be present in highly variable abundances. Some will be at high abundance, some at low abundance. And we also have very large data sets. So we need scalable algorithms for metagenomics. And this is in a sort of a brief illustration of the problem. Here I've built a short read assembly graph out of 6 strains of the same species. And you can see the complexity of graphs that result. Now this motivated metagenome binners, which I believe we're going to have another talk about later on in this session. And the principle here is that, okay, so we end up with lots of these fragmented contigs, but if we can find features that all the contigs from the same genome share, then although we might not be able to assemble the genome, we can at least link all the contigs that derive from that genome together and get the full genomic information. And for doing that, we typically use 2 different sources of information: composition, which reflects the sequence of the contigs. And as I said, I think it's likely that a later talk will go into this in more detail. But an additional source of information that I've worked on a lot is coverage across multiple samples. So the principle here is that if we have multiple contigs from the same species, and that species varies in abundance across more than one sample, all those contigs will have the same pattern of coverage depth across samples. And I've illustrated this here by taking a set of contigs, forming coverage and composition vectors, and then doing a PCA. And you can see that we get these clusters in 2 dimensions. We devised Concoct as one of the first algorithms to use both sources of this information. And in that case, for clustering, we used a Gaussian mixture model. Now, when you've built your bins, you want to evaluate them. And here we'd use single copy core genes. These are sets of genes that we know are present in all prokaryotic genomes with just a single copy. So the fraction of those genes you observe gives you a sense of the completeness of the bin, and the number that are in multiple copies tell you about the contamination. Now, today, there's lots of such binners available, but they all broadly work on this principle. You transform the data and then use some form of clustering algorithm. Probably the big breakthrough is using deep learning methods to do this embedding in a far more sophisticated way than we did in the original PCA. So yeah, I am excited to hear about this binner later on today. Okay, so that's a brief introduction to short read metagenomics and genome-resolved metagenomics from short reads. I mean, what can we do with it? Now, there are major limitations. You only ever get a fraction of the sample with short reads. You can do better if, as I said, you've got multiple samples from the same community. But even then, you'll be missing things like 16S ribosomal RNA operons because they form large repeats. But it has been very successful, and it's been successful because if you have enough samples, even if you can only get a fraction of the community as bins, you can still build up a comprehensive collection. At least that works for the human gut. So this is nicely illustrated, say, by this paper by Pozzoli et al. They got 150,000 mags from 80,000 gut samples. It seems to work less well for other environments. So I was involved in a study of generating mags from the Tara Oceans data sets. And there, I think we were estimating we were getting about 15% of the genomes. There have been some recent studies building mags off soil, but I suspect that those are even less representative. Okay, so this motivates the use of long reads. They've got major advantages, as I will talk about. Now, in terms of the state of the art, we've had ONT reads for a little while, but you don't tend to get, from those older ONT reads, you're not getting great results in terms of, getting a comprehensive representation of the community. So from this Moss et al. Paper, they had 13 human stool samples. They did 1 minion per sample. That's about 10 gigs of data per minion. And they only got 20 circular genomes. Now, the first results from Hi-Fi came out in about 2022, and that was much more exciting, as I'll talk about. Now, in terms of methods for assembling Hi-Fi data, these long, accurate reads, you can't just use de Bruijn graphs, because de Bruijn graphs won't scale to long reads for 2 reasons. Firstly, we need long overlaps. So say, if you've got a 10,000 base pair read, you need, say, up to a 5,000 base pair overlap. Even with a k-mer length of 256, we've got 10 to the 154 different possible k-mers. So that k-mer complexity becomes very large, but also it's very bad at addressing noise, because any error on that k-mer will affect your ability to assemble. So most of the state-of-the-art long-read assemblers are using some form of string graph. That would be like Canoe or Hi-Fi as a meta, which is a derivative of the miniasm algorithm. Metafly, which I have to admit I don't understand fully, is using a combination of de Bruijn graphs followed by a repeat graph and read mapping. So I think they're using a sparse de Bruijn graph where you subsample k-mers to do an initial assembly to get small contigs, and then they're improving on that with their repeat graphs. Now, this motivated us to see whether we could produce a more scalable Hi-Fi assembler based on something called a minimizer de Bruijn graph, which I'll explain in a second. So I mentioned minimizers earlier. For our assembler, we use a concept called a universal minimizer. So this is quite simple. So if we consider our read, we decompose it into k-mers as before, but each k-mer is then randomly hashed to an integer. But that's a reproducible hashing. And then we set a threshold, and we keep all k-mers whose integers are below that threshold. So we're subsampling k-mers, but it's done in a reproducible fashion. And minimizers, as I said, are behind a huge number of bioinformatics algorithms at the moment. Now, the minimizer de Bruijn graph was introduced by Ryan Chickey in 2021. And the principle here is that you can build a de Bruijn graph out of anything. So you don't have to use k-mers. So instead, he proposed building a de Bruijn graph out of lists of minimizers. So now each node on our graph is not a nucleotide word, it's a list of those minimizers. Now this has multiple advantages in terms of the assembly. It's obviously far more efficient because in our case we're only using 1 in 200 of the k-mers, but it's also more robust to noise because if an error lies between minimizers, then you effectively ignore it. And in particular in metagenomics, it turns out to be very effective, I think, because you're effectively doing a lower resolution assembly because you're only using those minimizers. Okay, so we took this idea of Ryan's, and when I say we, I actually mean my postdoc Gaeton. He did the entire implementation of this program. He is now actually at the Pasteur Institute with Ryan. And he re-implemented it specifically for metagenomics. Now, the MetaMDBG, that's the name of his program, it has 3 parameters. The size of the minimizers, those k-mer lengths, the density of selected minimizers, which we fixed to be 0.5% of k-mers, so 1 minimizer every 200 base pairs, and the length of those k-mers, how many minimizers are actually in each of those nodes. Now, just as for a regular de Bruijn graph, you want long k-mers for high coverage organisms and short k-mers for low coverage organisms. So what Gaeton did was to devise a multi-k approach specifically for these minimizer de Bruijn graphs. And the really neat thing about it is that it all functions in minimizer space. So the first step is to convert reads into these minimizer lists. We call those M reads. He does some filtering. So this we would also do in a standard de Bruijn graph. He removes k-mers below a certain frequency, but he does this in an intelligent way so that it's all relative to the abundance of a read. He constructs the de Bruijn graph. He would then do standard simplification like tip clipping, bubble popping, and then he has his own novel algorithm, something called a local progressive abundance filter that I'll explain in a moment. That produces contigs in minimizer space, and those are then fed back into the next iteration of the assembler, along with the reads. And you repeat that from k-min to k-max to form your assembly. The progressive abundance filter is basically a way of also removing errors and strain variation, where you find a long starting contig, you determine its abundance, and then you progressively filter less abundant nodes. And in this way, you can actually separate out, you're sort of exploiting that variability present in metagenomics data to improve the quality of the overall assembly. So we did a very simple analysis pipeline to compare MetaMDBG to other assemblers. We perform the assembly, then actually we take all the circular contigs and they go straight to MAG evaluation. And then we do a binning with MetaBat2. In fact, I did encourage Gaeton to look at other binners such as our own, but he wanted to use what he considered to be the standard binner in the field at that point. And then we evaluate the bins. So we use checkm for that. We define near complete as greater than 90% completeness, less than 5% contamination. And then we've got circular contigs that are near complete that have come in straight from step 2, high quality, medium quality, etc. So how did we do? We evaluated it on 3 different data sets, a pooled human gut sample of about 60 gigs, an AD sample of 170 gigs, and a sheep rumen of over 200 gigs. Now, what was really impressive was in terms of the computational performance. On the larger, more complex data sets, we were between 5 to 10 times faster than HiFi and Meta, maybe somewhat less improvement compared to MetaFly, but we also did really well in terms of memory required. So on the complex sheep data set, we required half as much memory as MetaFly, I think a fifth of MetaFly and a tenth of HiFi and Meta. So that was good, but we also got better results. So in terms of number of mags, which I'm showing on the left here, the green bars are the number of these near complete circularized contigs. So it's basically a perfect genome from a metagenome. On the more complex AD data set, so AD, by the way, is an anaerobic digester. This is a biotechnology for producing biogas, and you can sort of view it as a more complex human gut. It'll have more species, more strains, etc. But we were getting twice as many near complete circularized contigs, and then more mags overall. Maybe less impressive on that lower coverage human gut. But another way you can evaluate this is in terms of the total fraction of reads mapping onto the mag collection. And there we did really well on the AD data set. I mean, we had nearly 40% of reads mapping onto circularized mags. And if you consider all mags, we were getting something like 70% of the community. So why did we do so well in terms of reads mapping? It's because we were doing a better job of assembling high coverage organisms. That's what I'm showing here. So this is the coverage depths of mags obtained by the 3 different assemblers. So with MetaMDBG, we're able to get more high coverage mags. And this is it's to do with this fact, I think, that we're actually effectively operating at a lower resolution. We're ignoring a lot of the complexity of strain diversity that happens when you have a high coverage organism. And yeah, that enables us, because those high coverage organisms, because you will see a lot of diversity in them, they cause problems to things like the string graph assemblers of HiFi and Meta. At least that was our intuition. In terms of the mag quality, for those mags that weren't circularized, most mags were just in 3 or 4 different contigs, which is really, an incredible level of quality, but that was similar across assemblers. So there's no major difference in terms of number of contigs in near complete non-circular mags. We also, I mentioned that with short reads, you can't get 16S sequences. We evaluated the mags in terms of the numbers of 5S, 16S, 23S, and the 18 tRNA genes that were observed. And yeah, 96% of mags from MetaMDBG had all of those ribosomal genes. And yeah, that was similar, although MetaFly did slightly better on that metric. We also got better recovery of phages and plasmids, but I won't go into that in detail. Now here I'm showing, just for that anaerobic digester data set, I'm showing the phylogenetic diversity of mags that were obtained. So each, on this phylogenetic tree, each node is a genre, and the outer layers are showing the number of mags obtained by each of the different assemblers. So you can see we've got large numbers of genre, which were only obtained with our assembler. There's a few that were only obtained with MetaFly, and I think just one that you could only get from HiFi and Meta. And that's an important point as well, because it sort of shows that although we would say that our assembler is looking better on these data sets, there are still organisms that you can't get from our assembler that you would get from the other assemblers. So it does suggest that if you wanted the most comprehensive assembly, you might try multiple assemblers and reconcile them, similar to what people do with binning, for instance. And yeah, this is just the statistics on those taxa numbers that were unique to the 3 different assemblers. Yeah, and we got 6 phyla that you would only see with MetaMDBG from this anaerobic digester data set. Okay, so that's nice, and people are always showing, how great their algorithm is. So now I'm going to show some results from somebody else using our algorithm to, hopefully convince you that it's not just us that gets good results out of it. So this is actually, so MetaMDBG was picked up by PacBio themselves, their bioinformatician there, Dan Portig, and he's used it in this recent study that's just appeared on the archive, and it's, I think, under review with genome biology. And there he sequenced this Zymo-Biomics reference standard. So this is a mixture of human gut samples, which may have undergone some enrichment, where they did really deep hi-fi sequencing. So they did 3 Sequel2 flow cells, 2 Revio flow cells, a total of 250 gigs of sequence data. They also did Hi-C for this sample, which I mentioned earlier, that proximity ligation. So he tried Hi-Fi as a meta and MetaMDBG. He has his own MAG pipeline, which I think is a combination of SemiBin2 and MetaBat2. But he also used the ProxyMeta MAG pipeline, which uses this proximity ligation. He compared bins between the two, generated taxonomies, etc. And actually, this entire pipeline is available on the PacBio GitHub. So what were his results like? Well, actually, he did get overall slightly more mags with Hi-Fi as a meta, but from MetaMDBG, he was getting more high-quality mags. He also got somewhat more mags using the Hi-C information, but it's not dramatic. And yeah, the best results were a consolidation of that Hi-C information and the what he calls bioinformatics binning. This is showing the number of mags as a function of dataset size, which is intriguing. So we do start to see these curves saturating. So it is suggesting that actually we are getting close to resolving all the mags that are present. We can get a sense of that, though, in 2 ways. One is, again, to look at the fraction of reads mapping onto the mag collection, and that was about 60%. Another way to do this is to look at the single copy core gene number in the assembly. So if we take each of those single copy core genes and see how many different versions we observe and compute the median across our collection, that's an actual estimate of the total diversity in the assembly, which came out at about 750. So comparing that to the number of mags obtained, it's a similar sort of number. So it looks like we're getting between 65 to 70% of the community from a single sample as high quality mags, which is really dramatic. But there is a question of how this would do on more complex community. And here I'm also showing you some more of Dan's data. This is from two different soil samples, where again, they've done deep hi-fi sequencing. And it's pleasing from our perspective, because you can see that we get more mags out of MetaMDBG than hi-fi as a meta. And I think this is possibly going to be a feature of our algorithm going forward, that will do better on these more complex communities. But the amazing thing here is that you are getting hundreds of mags directly out of soil, which I believe has never been done before. And also note how these curves are not saturating at all. So it does suggest we would have to do a massive amount of high accuracy long reads to actually get a representative sample from a soil community. So I think that's pretty interesting. So just briefly in terms of future outlook for ONT data. So it's clear hi-fi has huge potential, but it is expensive. ONT error rates, they're getting a lot better. It's around about 0.5% now, and they are cheaper. So it's very appealing to think about Oxford Nanopore. In our paper, we did use ONT reads after first polishing them with short reads. And in that case, actually, we got pretty good results compared to Metafly, which is the state of the art for Nanopore, I would say. And noticeably, we got more circularized mags, but slightly less mags overall again. But what Gaeton's working on now is a dedicated ONT version of MetaMDBG. And what we hope here is that this will be something that is scalable to large nanopore data sets, which is probably what we need to understand complex environments. Okay, so to summarize then, I think, at least for low complexity and intermediate complexity environments, things like the gut and those anaerobic digesters, high accuracy reads really do allow us to get the majority of the community as high quality genomes. We've demonstrated we can do this in a scalable manner with minimizer to Bruin graphs. This may be true of more complex communities if you can afford to do all that sequencing, but ONT could well be a promising alternative. Although it's always worth bearing in mind, and this also relates to another talk later in the session, that when we build a MAG in this way, we're going to still have population variability. So having built the MAG, you want to try and resolve the different strains that are present. And here programs like, we worked on one called Strainberry, Mihai has also worked on one called Strainy, and then Floria, I think, is later in the session doing this with noisier ONT reads. So that is exciting. And combining these high quality assemblers with those strain resolution algorithms, yeah, we could really get the entirety of a picture of the community. If this interests you, we will be recruiting PDRAs at the Erlen. And yeah, you can just send me an email. One thing to bear in mind though is we're putting all this effort into long reads, but this is something I asked one of my postdocs to do. So this is the JGI gold standard data sets. It's still, the fact is there's 35,000 fecal samples from Illumina metagenomics, and there's only 36 PacBio and 60 nanopore. I was actually quite surprised at how few nanopore there are. But yeah, so the question is, do we really need these tools if nobody's actually doing long read sequencing, which is something to think about. But anyway, so I'll acknowledge in particular Ryan, Gaeton, who wrote the assembler, Ryan, who his initial idea this was, but also Seb doing the bioinformatics in my group, and Rob that prepared all the samples, but also Dan at PacBio, and then the BBSRC. So thank you very much.
So we have time for a few questions. If you have a question, can you come up to the microphones? I have one of them.
Thank you. That was really interesting. Thanks. I was wondering, you have these variable K methods where you vary the K-mer size and repeat. Can you also do that like varying the hash? And so it's still deterministic, but you could get different, in case you get unlucky with, there's some like variant in what you're downsampling to.
Yeah, that's an interesting question. And actually, it's a good question because somebody else asked me this very same question last time I gave this talk. And yeah, there's no doubt you could try that. And I think it's possible, you could repeat the whole assembly process with a different hash, and you may well get some things that assemble better. The other thing we did think about was whether you could vary the density of the minimizers, but you certainly can't do that in the process within the same graph. That would vary the graph. But yeah, you could definitely try a different hash, and possibly more in, the thing is, I suppose it would work with any way of building the minimizers. So there are more intelligent, I believe, functions for actually doing the minimizers that you could think about. But yeah, for sure, that is possible to worth trying.
Cool, thank you.
Other questions? Is there anybody online that's asking anything? Just make sure we're not missing them. No questions online. Chris, I have a question, which is the long read technologies do require high quality DNA. So the question is, do you get a sense of what's missing from the community that perhaps doesn't glyze properly and then gets fragmented, doesn't end up in the data?
Yeah, that's also a good question, Mihai, and it's one, because we're doing the sequencing ourselves in a lot of these studies, it's one we're very, very aware of. To build a PacBio library, you need incredibly high quality DNA. So you don't just need long fragments, but it mustn't have any nicks in it. So you need a really good quality sample to begin with. So having done that, I think you can come up with extraction methods that are representative. So I don't think we're missing too much of the community. I just think it's just more of